and discuss how the UK and ASEAN can work together in genomic medicine. I'm Alan Lai with the UK-ASEAN Business Council based in Brunei and have the pleasure of being your MC for today. Before we get underway, I have three things for you to note. Firstly, in addition to our cast of wonderful speakers, it is important that we also hear from all of you taking part. So please do share your thoughts in the chat box, and also during the panel discussion, please do place any questions you may have in the Q&A box. Also to note, this forum is being recorded and a copy will be available via the UK-ASEAN Business Council's website and on our YouTube channel. And thirdly, if you are on Twitter, do tag us at UK-ASEAN. To get us underway, it is my absolute pleasure to hand over to His Excellency John Lam, the UK's Ambassador to ASEAN based in Jakarta. Ambassador Lam, good to see you again. Over to you.
Well, many thanks, Alan, and thank you all for joining us today. Now, as Alan says, I'm the UK Ambassador to ASEAN based in Jakarta, and it's really a great pleasure to welcome you and to introduce the symposium today with so many partners from across the ASEAN region and the UK. Now, this is a webinar jointly hosted by the UK government and by our friends and partners at the UK-ASEAN Business Council, both of us, of course, committed to promoting economic opportunities and deeper economic links between the UK and Southeast Asia. I would also like to thank the ASEAN Business Advisory Council and the Ministry of Health Malaysia and My Digital as co-partners today. And I'd really like to thank the keynote speakers who are joining us from both the UK and ASEAN sides. It's a fantastic group of speakers with a huge range of expertise, and I feel particularly lucky that I'm going to be able to listen and learn from them all today. So not only are they fantastic experts in their field, but they are driving innovation right across the field of genomics. And from a UK government perspective, we're really keen to generate discussions and partnerships that will help to support, strengthen and develop the economic links, the mutual prosperity that I spoke about earlier. But why have we chosen genomics as the theme today? You probably don't need me to tell you, but genomic medicine has already had huge impacts in improving health outcomes globally. It provides incredible opportunities for more accurate diagnoses, faster treatment, and is driving a revolutionary shift towards more personalized medicine. So that means more targeted treatments for patients with diseases such as cancer, obesity, and cardiovascular disease. And I think it's something, at least for me, that has become even more evident and obvious as we've been tackling the COVID-19 pandemic, where we've seen the vital role that genomic sequencing has in terms of protecting our global health security. And that's a huge priority for us in this region, as I'll come on to in a moment. Before I do that, I'd like to say a word about what the UK brings here. You know, we are really proud to be home to a world-class healthcare and life sciences ecosystem. We have two of the top 3 universities in the world for life sciences and a £74 billion turnover in the wider sector. And along with that wider infrastructure, the UK is proud to be a global leader in building an end-to-end ecosystem that supports and accelerates the power of genomics. For example, Genomics England achieved the 100,000 genome sequencing project in 2018, and the UK was the first country to apply the whole genome sequencing at scale to our system. Looking to the future, the UK government launched our new Genome UK implementation plan in May 2021, and that sets out 27 commitments to deliver better technology, faster testing, and again, delivering whole genome sequencing in our health system. More recently, we launched the UK Life Sciences Vision, and that's building on our clinical research, genomic and health data capabilities. So taken all together, that supports a £5 billion sector in the UK, employing over 5,000 skilled people in more than 150 companies. But we can do a lot together. We can learn a lot from our partners here in ASEAN, and we can work together on shared projects. So right across the ASEAN region, there's a range of innovative institutions, universities and private sector companies looking to use genomics to improve health outcomes in this region. And we're keen to understand how we can collaborate from research, developing new ideas, and also putting them into practice, commercializing them in business. We feel that by working in partnership, we can find new opportunities to develop the next generation of life-changing treatments and technologies, all the while securing our health systems from future hazards. Now, the UK is already doing a lot. The UK government supports the ASEAN region with initiatives under our Fleming Fund and our Better Health programme that we deliver in a number of ASEAN countries. As an example, in the latter programme, we're working with the Thai government to support the Thai healthcare system on genomics education and training, as well as wider genomics research partnerships. And that's just a subset of our wider health cooperation with the ASEAN region, which includes, of course, a huge amount of cooperation on tackling the COVID-19 pandemic and recovering from it together. So what happens next? And this is really what today is all about: identifying the new opportunities, thinking about new ways where the UK and ASEAN work together, and thinking about how we can use those to kind of build back better, recover from the pandemic. So we really hope that today's event facilitates new insights, and it's my pleasure to hand back to Alan to take us forward through the rest of the webinar. Thank you again.
Thank you very much, Ambassador Lam. And now I am delighted to hand over to the chair of the ASEAN Business Advisory Council's Healthcare Working Group, Dr. Kaw Swee Keng, to deliver his opening address on behalf of the Council. Dr. Kaw, over to you.
Thank you, Alan. On behalf of the ASEAN Business Advisory Council, it's a pleasure for us to be a partner, and we welcome you to the Genomic Symposium organized by the UK government and the UK-ASEAN Business Council. Allow me to make my opening remarks in two components, Alan and my friends. The first one is to sketch out the history of drug development over the last 120 years or so, and then secondly, to bring us to the present in Southeast Asia. When we're talking about the story of medicine's development, we can probably define it into three phases. The first phase started with aspirin that was approved in the year 1899 and culminated in the peak sales of Lipitor or atorvastatin, which is considered a small molecule, at about $13 billion a year in around the mid-2000s or so. That's the first phase, small molecules, which are easy to engineer and very widely available. The second phase of drug innovations is actually within us right now, which is the biologics phase. For example, the medicine called Humira or adalimumab achieved peak sales of $20 billion a year. And biologics are much more complicated to manufacture because they are larger molecular weights and they have very complex manufacturing, but they also have tremendous business opportunity, witnessed by $250 billion worth of biologics being purchased every year, as well as 8 of the top 10 best-selling drugs in the world being biologics today. That's the second phase, biologics. Right now, we're starting the third phase of drug discovery, of genomics, of precision medicine, of understanding the human genomes, not so much the proteins, but also the human genomes and that gives the instructions for these proteins and so on. So as we embark on this genomic phase of drug discovery, it is fascinating to see that we've come a long way in only 120 years. Alan and my friends, let me move to the second part of my comments, which is about how genomics and how from small molecules to biologics to genomics, how will that impact Southeast Asia and ASEAN? ASEAN is 650 million people with an economy of approximately $4.5 trillion. And there are 5 reasons perhaps that the partnership with the United Kingdom, for the reasons that Ambassador Lam have mentioned earlier, will be able to have a great connection with Southeast Asia. These are the 5. Firstly, there is a long-standing relationship between the United Kingdom and countries in Southeast Asia, diplomatically, trade-wise, and for a certain period of time, even militarily speaking. And I think the educational, scientific, and trade links between our countries and regions will do well for us in this collaboration for genomics and genomic medicine. That's the first one. The second reason for why Southeast Asia is because there is significant human capital in Southeast Asia, especially highly educated, highly qualified, internationally trained human capital that we can tap on in order to expand the genomics market as well as R&D. 650 million people, for example, is almost 10 times the size of the United Kingdom. Thirdly, there is significant R&D infrastructure in the universities, the private sector, as well as the research institutes in Southeast Asia. And I'm glad that many of them are represented here today. And if we're able to harness the existing R&D that's inside Southeast Asia and ally them to the cutting edge innovation that's happening in the UK and other countries around the world, I think this will be a very powerful combination. And that's the third reason why this partnership can be very successful. And the fourth reason is that Southeast Asia is increasingly capable of high-end manufacturing. So not just For example, during the COVID pandemic, where we're able to fill and finish vaccines, but also to manufacture and also to research from scratch COVID vaccines and various other paraphernalia that is needed for us to combat the COVID pandemic and other public health threats. And the final reason is obviously a market of 650 million people with a rapidly increasing middle class. And for these 5 reasons, I'm confident about the potential successes of UK and ASEAN's collaboration in genomics. Alan, I'll pause here and end really by saying congratulations again to the UK-ASEAN Business Council and the UK government in organizing this important symposia, and we wish all the participants a great symposia. Thank you very much again, Alan, and over to you.
Dr. Kaur, I've learned so much in 5 minutes in the last 120 years. So that was a brilliant opening address. Thank you very much. And that indeed sets us up nicely for our 2 following keynote addresses, which we have 2 very important addresses that will paint a picture of genomics in both the UK and in ASEAN. To begin, I'd like to invite Sir John Byrne, Professor of Clinical Genetics at Newcastle University and Chairman of the Newcastle upon Tyne Hospital's NHS Foundation Trust and Vice Chair for Quantu MDNX Limited. Professor Sir John Byrne, the floor is yours.
Thank you, John, and hello, everyone. Mr. Ambassador, ladies and gentlemen, it's a great honor and a pleasure. Could I have my first slide, please? So I've taken as my title, Genomic Medicine, and I'd like in the next 5 minutes to give you a brief overview and also 3 examples of the work that we're doing in this space with our colleagues in Asia. Next slide. So we've been involved in the UK in genomics since its inception at the beginning of the 20th century. Fred Sanger won his second Nobel Prize for working out how to sequence DNA at the base of the Human Genome Project. Shankar Balasubramanian invented the next-gen sequencing technology, which gave us now the NovaSeq. We have one in Newcastle, which will do 48 whole genome sequences per run over a 2-day period. Bearing in mind, it took us 12 years to do the first one, and it cost about £4 billion. Sorry, that was animated to get us quickly through. Genomics, I helped to write the report with John Bell, which prompted David Cameron to create Genomics England, and I still sit on its scientific advisory committee. And by 2019, we were up to 116,000 genomes and still going fast. And we are inventing new ways of using that technology. Not least, of course, Nanopore, which was recently launched for several £1 billion under the markets with its long read sequencing technology. And then finally there, the picture in the bottom is our 7 genomic laboratory hubs. We lead one of them from the north of England, covering 8.5 million people, delivering next generation technology into the NHS at scale and at speed. Next slide. So at the other end of the market, Quantum DX is a company that we've led from Newcastle for about 12 years now, and we've developed a point of care PCR device, which is battery operable. You can carry it in a backpack on your back. And this is partly done with our colleagues in Singapore. We use technology at the A-Star laboratories in its early stages. And we now use Univac in Singapore to manufacture the disposable cassettes. We introduced our Prime Minister to the device, which has 1,000 working parts last year and is now licensed for use in our clinics. And it's worth pointing out that as we develop genomic medicine for the 650 million people of South Asia, we can't just use centralized laboratories. And so these devices can be used to take the DNA testing out to people. We first, of course, have done COVID-19, and we can give you a PCR result at your bedside or at your dining table. And I took it to dinner with Kate Bingham and gave her a PCR test while we were waiting for our starter. So this is the future of genomics as it reaches out to every extent of our population. Next slide. And it's not just about the diseases of the West, it's also the diseases which are inherent in the Southeast Asian population. I co-lead the Global Variome at UNESCO, and we had this conference before COVID, needless to say, as you can see, of colleagues from across Southeast Asia looking at thalassemia. These are the 24 common variants in thalassemia, which became common in Asia because of malaria, and which now cause a huge medical burden. And we've been working with our colleagues in Malaysia and have a grant from the Malaysian government to develop a cassette for our device that will diagnose all 24 and be able to be used in Malaysia in their screening program for all 16-year-olds to avoid having to take blood from them. You can do it on a mouth swab. Next slide. We've also, of course, in the UK, been world leaders in COVID, as you've already heard mentioned, and just one small project, which has now been confirmed by our colleagues at Genomics England. In Newcastle, we looked at people who had severe disease who shouldn't have had, and compared to them those who had very little disease, even though one would expect them to be ill. And we've identified one specific variant in tissue type, the HLA-DRB10401, which is associated with resistance to having severe COVID. And this is one of about 20 markers now, which we've now identified in the genome, which explain why some of us go down with it and others don't. This is particularly important one, because if you look on the next, click again, if you look across the planet, you'll see that this particular tissue type it gets progressively common the further you get away from the equator. Almost certainly it's been selected up, just as the thalassemia markers were selected by malaria. This marker will have been selected by previous waves of coronaviruses and their relatives, meaning that people in the northern latitudes are more resistant to this coronavirus than people who derive from nearer the equator. Now, this is going to be very important in the future as it becomes endemic, to try and target our vaccines and to try and be more personalized in working out who needs to be protected and who doesn't. Next slide. The other major intervention over the last decade or so is the Global Alliance for Genomics and Health, which I am involved in and is now chaired by Ewan Burney at the EBI, the European Bioinformatics Institute in Cambridge. And when it was launched, I put forward the idea that we should gather all the people in the world to tell us what are the spelling mistakes in BRCA1 and BRCA2 that cause breast cancer and ovarian cancer. Stephen Chanik, who leads the National Institute of Health cancer team in the US, and myself co-chair this, and we had a meeting to launch it at UNESCO again. And the next slide shows the result of that initiative. And what we've actually now done is managed to create a free website which contains, next, just click again, 64,000 variants in these two genes, so that you can actually look up your variant. Next slide, next click. And in fact, we have an app so you can follow your own variant to see whether it actually is a pathogenic cause of cancer or not. And this data is not just from the West, but also we just recently included all the data from Japan. And so we're basically creating a worldwide resource to assist diagnosticians in genomics to work out who should be given particular attention to prevent them getting cancer of the breast and ovary. And of course, to target those new drugs that you just heard about. Next slide. So that's my 5 minutes of fame. I hope that was interesting. So the UK enjoys a rich tradition of collaborative genomic research. Combining our skills and experience and resources with those of partners in Asia offers rich rewards. And I hope I've given you a couple of examples of that. And understanding diversity can bring benefit to all mankind. Thank you for your attention.
Thank you very much, Professor John.
Start this up in your video.
And now to hear the ASEAN side and to understand the genomic landscape in Southeast Asia, I'm delighted to welcome Dr. Nurlisa Muhammad Nordin, focal point for strengthening the laboratory capacity initiative at the Ministry of Health in Malaysia. Dr. Nurlisa, over to you.
Yes, I can have, okay, thank you very much, Mr. Chamberlain. Bismillahirrahmanirrahim. Honorable representatives from the UK Department for International Trade in conjunction with Foreign, Commonwealth and Development Office, delegates from ASEAN member states, and distinguished all participants, a very good afternoon from Kuala Lumpur, Malaysia. On behalf of the ASEAN health sector, I would like to express our deep appreciation for the opportunity in providing information and updates on our 2021 to 2025 health sector priorities, specifically in biogenomics initiatives in our efforts in strengthening laboratory capacity of the ASEAN member states. Next slide, please. Allow me to briefly provide you a visualization as shown here in this slide that the ASEAN post-2015 Health Development Agenda of 2021 to 2025 with its specific vision, mission, goals, strategies, and implementation mechanism that are aligned and complementary with its existing regional and global commitments, agreements, and trends such as the health-related sustainable development goals and the health-related strategic measures of the ASEAN Socio-Cultural Community Blueprint 2016 to 2025. The ASEAN Post-2015 Health Development Agenda has 4 ASEAN health clusters, as you can see here, with their respective work program that consists of health priorities and project activities to be implemented from 2021 to 2025. The initiatives on strengthening laboratory capacity, including activities on genomic surveillance, are part of the ASEAN Health Cluster 2 on responding all hazards and emerging threats. We will deep dive into the biogenomic initiatives in the ASEAN as part of the ASEAN Health Cluster 2 program in the succeeding slides. Next, please. One of the major milestones in biogenomics initiative in ASEAN transpired from the directions of the ASEAN Health Ministers meeting in 2021, where all ASEAN health ministers agreed to enhance laboratory capacity across the region and enforcing genomic surveillance of SARS-CoV-2 to be able to swiftly detect and share information on potential variant of interest and variant of concerns of SARS-CoV-2. Based on the direction given by the health ministers through their joint statement on 22nd July last year, the ASEAN Health Cluster 2 and the operational body of the ASEAN health sector included specific project activities on capacity building and networking on biogenomic surveillance among ASEAN member states, with the targeted output projected in these slides. Malaysia, through the Ministry of Health, is the lead country for the COVID-19 biogenomic surveillance initiative, which is currently ongoing. Next slide, please. Next slide, please. There's one slide on the, this one. Yeah. This is my last slide where I wish to present to you on the ongoing project on strengthening laboratory capacity on COVID-19 biogenomics for the ASEAN member states through a hands-on training and networking activities. This implements the direction set by the ASEAN health ministers. These endorsed projects commenced in 2021 with the preparation phase, which consists of consultation and assessment activities. Recently, an orientation meeting was conducted with designated representatives for the ASEAN member states to lay out all the project activities and detail implementation plans for the training as well as hands-on activities. This project is being funded by Germany through the GIZ office in Thailand. As you can see in this slide, we aim to strengthen the laboratory capability and capacity of the ASEAN member states to generate COVID-19 genomic data in real time, to understand the outbreak and monitor transmission and possible future pathogen threats. In our training program, we will also invite experts and professionals from other regions to provide their expertise and experiences in COVID-19 biogenomic activities. Having participated in this important symposium is also to create a wider opportunity for future engagement and collaboration with the ASEAN health sector in the field of biogenomics and relevant laboratory strengthening activities. We do hope to expand our network for future technical exchanges and sharing of lessons learned from and experiences. Next, please. For further information, kindly refer to ASEAN website, as you can see here. With that, I conclude my presentation and thank you so much for your kind attention. Thank you.
Dr. Nurliza, terima kasih. Thank you very much for that.
Thank you. Sama-sama.
Selamat petang. And with that understanding of the genomics landscape in the UK and ASEAN, we turn now to 4 case studies to hear practical examples of what has taken place. Each presenter will have 5 minutes to present. The first case study from the UK is by Lina Inamdar, Program Lead, New Variant Assessment Platform at the UK Health Security Agency. Lina, over to you.
Thank you very much, Alan. And hello, everyone, and welcome to all delegates and distinguished guests. I'm really privileged to be able to present a small case study on the new variant assessment platform, which is the UK's global genomic sequencing and surveillance offer that is led by the UK Health Security Agency. Genomic surveillance is transforming public health action by providing a deeper understanding of the pathogens, their evolution, their circulation. Used with clinical, epidemiological, and other multi-source data, genomic data for pathogens with pandemic and epidemic potential informs risk assessments and can help support development of vaccines, therapeutics, diagnostics assays, and decisions on public health social measures. New technologies in sequencing and bioinformatics have emerged in recent years, and some countries have made major strides in establishing genomic capabilities and capacities. Whole genome sequencing increases the sensitivity of our surveillance systems and helps answer questions that otherwise we would not have been able to answer in a timely manner. So next slide, please. The new way, sorry, can you just go back, please? Yeah, thanks. So the new variant assessment platform, it is the UK's flagship initiative, which was established in April 2021 to strengthen the global genomic sequencing and surveillance, and it is a key UK government commitment. It aims to help countries that have limited genomic sequencing and surveillance capacities and capabilities. And we are operating in selected countries and regions and offering to sequence the samples in the UK if there is absolutely no capacity within the country, or providing technical assistance to countries and regions indeed to send in regional hubs if they wish to do so. NVAP, through this initiative, is contributing to addressing the global genomic sequencing gaps and timely action on variants that poses a challenge to our pandemic response. NVAP provides support in several ways, including through access to genomic sequencing capacity in the UK. For example, in the context of the Omicron surge, we offered rapid sequencing support to some of the South African countries, and we are supporting in our phase 1 of the program countries like Nigeria, Kenya, Pakistan, Brazil to increase their in-country genomic sequencing capacity and capability in the longer term through provision of equipment, sequencing reagents, training, and technical expertise. Next slide, please. So NVAP has already offered tailored program of support in phase 1 to 5 countries and 2 regions, that is Africa CDC, and we are collaborating with Singapore as well for some regional offer of support. And in phase 2, we are largely working with the WHO regional offices now to provide that support to a larger footprint. Through NVAP, countries have access to UK's expertise and advice on bioinformatics. You can see the different sort of work streams here. So we provide access to the bioinformatics expertise, risk assessment, variant and virus characterization. And there are different work streams such as I mentioned, the provision of the UK genomic service, enhancement of the local and regional hubs where we provide targeted support to countries through procurement of reagents, sequencers, equipment, technical assistance, mentorship, and also structured training. We have a work stream on informatics where we also support and provide mentorship for the ICT computational infrastructure, analytical protocols, pipeline development, and also the risk assessment, variant decision making, how to translate the evidence to public health sort of policy making. The other work stream that we have is also biological assessment of variants of concern, and many countries have told us that they have very limited capacity or expertise in terms of the functional assays, the neutralization assays, where we can study the impact on vaccines, drugs, and therapeutics. And in the UK-HSA, we have a WHO CEPI-led platform, which is called the Agility Platform, and through that we are providing that technical assistance to countries. So briefly, I wanted to, in the next slide, mention about the NVAP achievements. And in a very short frame of time, we have established an end-to-end pathway for SARS-CoV-2 genomic sequencing, which we have the ambition to move to pathogen agnostic sequencing in the future strategy. We've helped countries to improve their sequencing capability. More in terms of practical support, we have provided support for protocol development, tools, resource development, worked with countries, to do the scoping, develop minimum data sets and protocols for data sharing, bioinformatics, wet lab sequencing, as I mentioned about the rapid support as well to the South African countries. One of the key challenges, as I'm sure colleagues here will identify, is negotiating data sharing, which is key for better pandemic preparedness, and also material transfer agreements. And we have negotiated that with our partner countries who have agreed to share genomic data on open platforms, and also with UK-ASEAN for rapid risk assessment. We've also engaged with a number of partners internationally. As you can see here, the WHO, we are working very, very closely with, but also National Public Health Institutes, the laboratories, Gates Foundation, and Rockefeller, we have engaged with to ensure that there is no duplication of efforts. So next slide, please. So in the, I just wanted to illustrate, this is not the latest slide, but latest map, but through the NVAP program, we are providing global reach to countries, who want to improve their genomic sequencing and surveillance. We know that the pandemic has really exposed challenges for genomics in the public health surveillance systems. The laboratories and networks previously dedicated to disease-specific threats such as influenza, TB, measles, polio, they were all mobilized to support SARS-CoV-2 genomic sequencing efforts. In some contexts, though, the repurposing capacities came at a cost to disease programs at their own surveillance and diagnostic needs. In addition, the scale, the geographic representativeness, timeliness, quality, comparability, and integration of genomic surveillance outputs with epidemiological and clinical surveillance findings still remains weak. So we must urgently build the global genomic and epidemiological surveillance networks to rapidly share data for our better pandemic preparedness in future. And next slide, which is my last slide. So just looking to the future, we are now at an important nexus. The International Health Regulations Emergency Committee for COVID-19 recommended increasing the global sequencing capacities, encouraging rapid sharing of data, including the metadata, and for WHO to actively support countries to strengthen their genomic surveillance. Whilst recognizing its specialized contribution, genomic surveillance comes at a cost, and the cost is very high in terms of technical, financial cost, especially in resource-constrained settings. And therefore, as public health authorities, we must carefully consider the value added of such an investment, including the broader surveillance and public health landscape. So I just leave you with the thought in terms of questions, really, in terms of how should countries, that have developed the genomic surveillance for SARS-CoV-2, that could be strengthened in a cross-cutting way to serve the rubric of current disease programs needs, as well as for future public health threats. How can this be done in a way that is forward-thinking, coherent, and cognizant of the existing initiatives so that we have integrated and interoperable systems? And I'll stop there. Thank you.
Thank you so much, Lina. That was a real tour de force. And certainly some of those points that you raised could be taken up by the panel that we've got that will come on shortly. But we've still got, we've got a couple more case studies. We'd like now to invite Slave Pejovsky, the Vice President and Head of Genome Analytics and Bioinformatics for AstraZeneca's Center for Genomics Research. Slave, over to you.
Thank you, Alan. And also like to take the opportunity to thank the organizers to give me the invitation and ability to introduce AstraZeneca's Global Genomics Initiative that is led out of AstraZeneca Center for Genomics Research. I'll aim to save some time in this section, as I will be back later on to moderate the panel session as well. But if I could just introduce myself here, then that would save us some time later as well. So I'm the Vice President and the head of this Genomics Center. For today, I have just a single slide, which is on the next slide, that outlines our company-wide Global Genomics Initiative strategy that was set out in 2016. So the strategy that we formulated back in 2016 was really focused around 5 key components with 4 primary goals to derive out of this genomics data. So first one was a bold ambition to analyze the sequences of up to 2 million individuals, including samples from our very own clinical trials. And what we'd like to do is integrate this genomic data that we get on these participants with other rich omics data, transcriptomics, proteomics, metabolomics, radiomics, and clinical data to understand disease outcomes, prognosis, response, and so forth. When we set out, we intentionally defined the strategy to focus our internal capability build around building the bioinformatics and the informatics infrastructure required to handle such large volumes of genomics and clinical data, and also the advanced analytical capabilities required to mine insights from this rich and high volume of data. Our sequencing, our data generation capacities, we formed strong collaborations with sequencing providers. So we send the specimens to them and then we get the sequence data back and we focus on the insights. We also focus on next generation sequencing. And this is to help us also detect the rare genetic variants that are often missed in the traditional microarray genotyping assays. And a primary kind of motivation behind this is that we'd like to focus on variants, genetic variants in the population that have high clinical effect sizes. And it's well known that rarer variants tend to have larger effect sizes just by the nature of the population genetic forces that act upon genetic variants. And so this is not to say we're not interested in common variants, but with the ability to sequence these individuals, we can also capture these rarer variants that have higher effect sizes, with our belief that identifying genetic variants and genes that are impacted with high clinical effects, this could actually translate to targets that, when perturbed therapeutically, may have stronger clinical efficacy than genetic variants that have very low effect sizes in the population. And finally, and perhaps most importantly, and very appropriate for this setting, is that when we set out on our strategy, we knew that we wouldn't be able to achieve this alone. And so we intentionally sought out to define a strategy that is outward facing, and we delivered this strategy through global academic and industry collaborations with experts across the world. The 4 areas that we prioritize on, and there's a lot more that goes behind each one of these, but at a high level, and probably very familiar to a lot of people on this call, we'd like to use the genomics data linked with clinical and outcomes data to better understand the underlying disease biology of many of the diseases that inflict humans. Next, we want to identify new targets for tomorrow's medicines, particularly targets and medicines for diseases with large unmet clinical needs. We want to use genomics to support selection of patients for our clinical trials on the basis of the molecular cause of disease, rather than perhaps the traditional paradigm of focusing on just the clinical presentation of disease. And related, we want to develop medicines and be able to allow our patients to be matched with the treatments that are most likely to benefit them. And we've heard a bit about this from our previous speakers. It's this precision medicine component, and this is an increasingly important component in our drug development pipeline in terms of developing medicines and companion diagnostics that match patients to the medicines that will most likely benefit. And I think overall, we're starting to see a bit of a paradigm shift in research and clinical trials and in all genomics aspects, in that, historically, when we've grouped people based on solely clinical presentation, we're now, with these genomics and other omics techniques, able to molecularly diagnose individuals and then identify different causes of the same clinical presentation. And so each of these different causes may benefit from different medicines. And this is a key part of our strategy going forward. So I'll finish up there. And like I promised, I'll be back to moderate the panel session in a few minutes' time. Thank you, Alan.
Thank you so much, Lava, and thank you for keeping to time. We really do appreciate that, and look forward to welcoming you back shortly to moderate the panel. We turn now to Southeast Asia, and our next presenter is from the Philippines. I'm delighted to welcome Cynthia Saloma, the Executive Director of the Genome Institute at the University of the Philippines System, Genomics Initiatives in the Philippines, for your 5-minute presentation. Over to you, Cynthia. Thank you.
Thank you very much, Alan. Can I have the PowerPoint presentation? I think it will begin. Okay, so you're not going to show my video. I actually have a 3-minute video about the Philippine Genome Center. I think that captures very well the initiatives that we have in the country. Can you have it played, please? Do you not have sound?
but more importantly, it serves the needs of the Filipino people.
The Philippine Genome Center is a genomics-focused and multidisciplinary institution which combines basic and applied research for the development of health and therapeutic products and services, improve the competitiveness of our agriculture and aquatic resources through research.
The Philippine Genome Center provides a full range of DNA sequencing services from high-throughput capillary sequencing to medium and high-throughput next-generation sequencing. This is available to researchers and students nationwide. The DNA sequencing core facility is a government investment in research and development that we should all take advantage of. The tools are here enabling us to pursue cutting-edge research.
Aside from nurturing aspiring researchers and young talents, the Philippine Genome Center fosters collaboration within the academe and research institutes in the government and private sectors around the country and in the international community.
PGC helps transform the generated complex data into useful information for biomedical scientists, biotechnologists, biologists, and other scientists.
This is made possible through bioinformatics services to efficiently process, analyze, curate, share, and archive massive amounts of genomic data.
These available services and trainings held by the Philippine Genome Center established various innovative products and paved the way for research outputs that are directly beneficial to the Philippines. Every living organism's structure and function is governed by a genetic program. Underlying this genetic program are the codes encoded in their DNA and RNA that essentially serve as instruction manual or blueprint for the entire organism. Genomics is making sense of this manual by integrating the complex data into practical information. As such, it has revolutionized and made valuable contributions in the fields of health, agriculture, biodiversity, ethnicity, and forensics. Researches that address the needs in health and medicine, agricultural productivity for food security, and for environmental sustainability for the benefit of the nation. Since its inception, the Philippine Genome Center has been involved in strengthening the academic and research infrastructure of the country.
PGC serves as a catalyst and an enabling environment to accelerate genomics research in the Philippines. We provide a mechanism by which our local researchers and clinicians as well can pursue research and clinical genomics at the cutting edge of science.
So thank you very much. May I continue with my PowerPoint presentation for the remaining 2 minutes of my talk? So thank you very much to the organizers for the invitation to be here with you today. Can I have the next slide, please? So in 2012, the Philippine Department of Science and Technology embarked on efforts to enhance our local capability in genomics and bioinformatics. There is a 7-year roadmap supposed to be in my slide. If you cannot say it, then I'll just mention it. That was crafted with the establishment of the first Philippine local DNA sequencing core facility within the University of the Philippines system. So the UP Philippine Genome Center was established in 2009 as a genomics-focused facility that utilizes the tools of genomics towards improvement in health, particularly in diagnostics and therapeutics, and to enhance our country's competitiveness in agriculture, fisheries, as well as forestry, and to support for R&D, our R&D efforts in biodiversity, ethnicity, and wildlife forensics research. Next slide, please. So we have different programs within the UPPGC. These are supported by our core facilities in sequencing and bioinformatics, biobanking, and of course in proteomics, which just opened last year. We have 3 core activities centering on offering technical services, training, and human resource development, as well as of course in research as listed here. Next slide, please. So when the pandemic started early on, we were involved in the field validation of a locally developed RT-PCR test kit on COVID-19. We also developed hands-on and online training modules and programs to expand our laboratory testing capacity in the country in coordination with our Department of Health here in the Philippines. Next slide, please. So we began to be actively involved in genomic biosurveillance around the time when the alpha variant was reported. Since then, we have sequenced about 25,000 whole genomes and have been responsible for the early detection of the P.3 or the former Theta variant. Our genomic biosurveillance work has helped inform our country's policies in border control and mobility restrictions. We have now expanded our whole genome sequencing capacity with the launch of sequencing services in our satellite facilities in the Visayas as well as in Mindanao. We are likewise involved in the sequencing arm of the various vaccine trials in the country. So the goal in the next few months is to integrate the country's genomic biosurveillance effort to include the 7 subnational laboratories in the country into a national network for genomic biosurveillance. So here I have listed some of the genomics initiatives that we have done so far. So the Philippine Genome Center is not working only for clinical genomics, but we are also involved in health, in agriculture, in fisheries, as well as in biodiversity research. So I think I'm going to end my presentation here. Thank you very much for your attention.
Thank you very much, Professor Cynthia Saloma, and for your wonderful presentation from the Philippine Genome Center. Up next, from the Ministry of Health of Malaysia, our next presenter before the panel discussion gets underway is Dr. Ravindran Thayan, Institute for Medical Research, National Institutes of Health at the Ministry of Health in Malaysia. Over to you, Dr. Ravi.
Thank you, Alan. First of all, I would like to thank the organizing committee for inviting me. This is the first slide. I'll be giving a perspective of the genomic surveillance that's done in Malaysia. Next slide, please. So Institute for Medical Research, in addition to the initiative that we did for SARS-CoV, being one of the centers that provides genomic sequencing, we also provide testing for COVID up with a capacity of 5,000 a day. And also one of the labs that accredited perform evaluation of diagnostic kits for COVID. And then we also introduced some genome phenotyping assay in points of entry because genome sequencing can take some time, but with VOC, presumptive VOC, we could actually quickly identify who have Omicron and so that public measures can be taken immediately. As well as we have involved in SARS-CoV-2 vaccine development as well as antiviral research and involved in a few of the studies, especially looking at the seroconversion studies involving healthcare workers. Next slide, please. So surveillance of COVID interests of our variant of interest or other variants started in February 2020 in the IMR. It's one way to characterize and understand circulating virus in Malaysia as well as those detected among points of entry. Also to understand the transmission of virus within classes in the community. By the end of 2020, genome sequencing became more important as variants of concern play their part and begin to emerge in other parts of the world as well, and also Malaysia. Next slide, please. So, and these are the criteria that we use to select for samples. This is basically looking at WHO's criteria, and I probably will give a bit of present a bit of an idea. Usually, it's representative samples from high positivity clusters, samples representing different geographical location in Malaysia, as well as different timelines of collection, samples from severe patients, especially with those with unusual manifestation, fatal cases, broad-in deaths, pediatric cases, adults with no comorbid. Samples from international points of entries because that's why we detected most of our VOC were actually at the point of entry. Samples from fully vaccinated healthcare workers with breakthrough infections. Samples from sentinel sites. We do have sentinel sites, SARI and ILI, and we do selectively, some of the samples were put for whole genome sequencing, as well as samples with spike gene target failure. Next slide, please. Next slide, please. So this is basically giving an overview of the capacity of our labs. There are actually 10 labs that can do whole genome sequencing, 8 in Peninsular Malaysia. Okay, oh, sorry. This so to expand further genome surveillance capacity in Malaysia, we know initially we had only about 200 per month, but we did engage with what we call as a Genomic Surveillance Consortium was established way back in August 2021, comprising members from Ministry of Health, universities, as well as Ministry of Science, Technology and Innovation, MGVI. Currently, there's 10 facilities in Malaysia that have capacity to perform up to 3,000 WGS per month, but of course, looking at what UK can do and other countries, so we also we would like to increase further. And there's also another pipeline to outsource some of the services to private labs. So we have sufficient funding for the next 2 years, and results are usually updated in memo information sources as well as GSite. Next slide, please. This will tell you about the location of all those 10 sites. 8 located in Peninsular Malaysia, mostly 6 in Klang Valley, and 1 each in states called Kelantan and Pahang, and 1 each in Sarawak and Sabah. So sample collection, and we also have the green dots, which will tell you the sample collection which is done all over Malaysia and referred to the designated WGS labs. The platforms used are usually, there are 2 types of platform mostly commonly used, Illumina and Nanopore. And the IMR also assisted in setting up genome sequencing services in public health laboratory in Kota Kinabalu with grants from JICA and MOH, expected to start in June 2022. Next slide, please. This is giving you an update of genome surveillance of SARS-CoV-2. This data was shared by MGVI, one of our partners. So looking back, what is happening in Malaysia is similar to what happens in other countries. So way back in September 2020, we had the 524 variant lineage, that followed by a quick temporarily alpha, then with beta, and delta had a very long duration, and it's all Omicron right now. Next slide, please. This is future direction of genome sequencing that we hope to expand further beyond SARS-CoV-2, which is including other targets, including pathogens of concern in tropical countries like Malaysia, dengue, chikungunya, influenza, respiratory viruses, HIV drug resistance, and other niche area that we would like to look is personalized medicine. I think this is going forward, where we would like to look into drug compatibility, risk factors for some non-communicable disease like cancer or diabetes, understand host genetics and pathogenesis, and resistance to therapeutic, because we know some of those drugs eventually develop therapeutic, and this is also into vaccine and therapeutic development. I think with that, I thank you for your attention.
Dr. Ravi, thank you so much. That was absolutely brilliant. Really appreciate that presentation, and great to see the wonderful work ongoing in Malaysia. Ladies and gentlemen, we turn now to the panel discussion on where and how can the UK collaborate with the ASEAN region. Your moderator is Slave Pedrovsky, Vice President and Head of Genome Analytics and Bioinformatics for AstraZeneca's Center for Genomics Research. Over to you, Slave, to introduce your panel and to lead the session.
Thank you, Alan, again. And as promised, I'm back in this additional role as moderator for today's panel session. As Alan introduced the topic, I think it is an important topic for us. We want to ensure we can share learnings from experiences across the private and public sectors, across the geographical regions of the globe. So I do hope this panel session will also identify some mutual interests and opportunities to build strong, sustainable collaborations between the Southeast Asia region and the UK. Just a reminder for members of the audience, you can engage in our discussion via the chat box, and then at the end of the fishbowl, we'll reserve at least 10 minutes to go through as many of the questions as the time permits. I'd now like to invite each of today's panelists just to give a brief introduction to themselves and their organization. And I suggest we begin with Professor Matt Brown.
Good morning and good afternoon to everybody over in Asia. So my name's Matt Brown. I'm the Chief Scientific Officer of Genomics England. Slave, do you want us to present our organisations as well at this point?
Just an introduction, yeah.
Yeah, okay. So Genomics England is a large government-owned company that's responsible for the development and provision of genomic services and development of those services and genomics research within the NHS. It was established in 2013 and it ran the iconic 100,000 Genomes project, which established the evidence base for the introduction of genomics, in especially whole genome sequencing, into clinical practice. We're involved in the provision of genomics within the NHS, and we double consent all of our patients who are participating in the study so that they're available for research as well, creating what's called the National Genomics Research Library. This is the world's largest research resource for rare diseases, with over 70,000 family members with rare diseases and whole genome sequence data, and also the world's largest cancer repository with over 18,000 cases with both tumour and normal sequencing. That library includes extensive clinical information, including the initial diagnosis, longitudinal health data, and a huge amount of additional data like digital histopathology, imaging, proteomics, and transcriptomics. We've also been very active in COVID research. So we've recently completed a COVID-19 whole genome sequencing study involving 56,000 people, which is about to be published in Nature this week, and identifies 16 new and many druggable targets for a severe COVID infection. The data that we produce in our National Genetics Research Library is available for academic and commercial partners to work with. We also have an extensive diversity program over the next 3 years, looking particularly at African ancestry and South and East Asian ancestry members of the UK population. I noticed in the chat there was some discussion about potential for collaboration with ASEAN partners, which will be something that will be valuable to draw out. And we're open to partnership with academics and commercial partners globally. I'll stop there. Thanks, Alan.
Impressive indeed. Thank you, Matt. So next, Dr. Mari Miyamoto, would you like to, oh, actually, I see there's an order on there. Maybe we go to Dr. Niranjan Nagarajan.
Thank you, Savi. Good morning and good afternoon, everyone. My name is Dr. Niranjan Nagarajan. I'm from the Genome Institute of Singapore, and we are part of the Agency for Science, Technology and Research in Singapore, which is a bit like the National Institutes of Health in the US. I'm also an associate professor at the School of Computing and the School of Medicine in the National University of Singapore. So at the Genome Institute of Singapore, our mission is to develop and leverage exciting advances in genomics and the analytical technologies that work around it for human health, economic, and societal benefits, and including some of the major challenges affecting all of us, such as infectious diseases, antimicrobial resistance, and climate change. The research in the Institute is in 4 broad areas. So population genetics and precision medicine. We are the custodians of the whole genome sequencing data that's part of the National Precision Medicine Program in Singapore, finishing a first phase with 10,000 individuals and moving on to 100,000 individual cohort. We are also heavily involved in developing new single cell and spatial omics assays to study regulatory biology, as well as epigenetic and epitranscriptomic technologies for both studying transcription and transcription regulation. We are also interested in looking at non-model organisms. It's not all about human health. So we have a domain in the area of genome architecture and design. And the goal here is really to be able to read and write DNA as a way to study non-model organisms. I will stop at there and be happy to discuss about how the Institute links to UK and ASEAN.
Well, it's a pleasure to have you on the panel. Next, if we can move on to Dr. Shairat Uthapipu.
Hi, good morning and good afternoon, everyone. So my name is Shairat Uthapipu, and so I'm the Deputy Chief Executive Officer at Thailand Center of Excellence for Life Sciences, T-Cells. So T-Cell is an public organization under the Ministry of Higher Education, Science, Research and Innovation of the Royal Thai government. So our role is to promote research and innovation to the real use and commercialization. So our vision is to promote life science industry to become top 10 economy with equitable access for Thailand. And our mission and the way we work on, so we work on to accelerate people, it's accelerate, talk to people and accelerate their innovation that they have in the lab or in their companies already. And then to do that, then we need to build a very good ecosystem for the development, as well as we have to connect and catalyzing people to the right people, like researchers and people in the companies who like to talk to investors or to connect to the public sectors, and that's our role to connect them together. And as well as what we are doing here in T-Cells, we are a part of Genomics Thailand project. And as maybe you have known about the Genomics Thailand project already, that we the government has funded this project started 2 years ago, and with something like 140 million US dollars, and we invest a lot for Genomics Thailand project to sequence 50,000 types. And this is the component of to know about the basic of genome of the Thai population. And for T cells, we promote the business, as I said, that after we have all the genome data, so we may have to make use of that. And T cell is the one that would look into what we can utilize them in the data we have, and then try to generate or nurture up or build the business on genomics also. And maybe we have many more topics we are working at TCell, like medical devices and drug discovery and drug development, and also natural products extract development to be a drug. And if you have time, I think I can promote you, tell you more about what we are doing at TCell.
Maybe we can work some of that into the responses to the panel session and the Q&A. Thank you. And we've got our last panelist as well, Dr. Mary Miyamoto.
Thank you, Saleb. Thank you for having me today, the panelist. My name is Mari Miyamoto. I'm the Oxford Nanopore Technology Commercial Director for the APAC and Japan. Oxford Nanopore Technology's goal is to bring the widest benefits to society through enabling the analysis of anything by anyone, anywhere. The company has developed a new technology, new generation of nanopore-based sensing technology that is currently used for real-time, high-performance, accessible, and scalable analysis of DNA and RNA. The technology is already used in more than 100 countries, and we are really focusing on expanding users in Southeast Asia. And it's being used to the understanding for the biology of the human, plants, animals, bacteria, viruses, and the environment, as well as to understand the diseases such as cancer. Oxford Nanopore Technologies also has the potential to provide a broader, high-impact, rapid insights in a number of areas, including healthcare, food, and agriculture. Oxford Nanopore devices works by sequencing DNA or RNA directly, and it can sequence anything from the short to ultra-long fragment of the DNA. This gives a truly comprehensive picture of the genome. A huge advantage of our technology is that data is streamed in real time and can enable rapid insights. The technology is also fully scalable from the pocket size to the ultra-high throughput devices. The smallest format is a Flongo. This can address the need for on-demand, rapid, smaller tests or experiment, and it can be used in the labs or even in the field. Then it comes to the pocket size Niranjan, which is a powerful and portable sequencing device that can deliver high volume of the long-read sequencing data. For projects requiring larger sample numbers, there is a benchtop devices, Niranjan Mark 1. This can run up to the 5 million of Flongo flow cell at a time on demand. And finally, the largest sequencing device is a PromethION. This is suited to the high-yield, high-throughput project that is specifically designed to offer on-demand use of up to 48 PromethION flow cells for use in core labs, academic institute, and for population-scale sequencing project alike. Thank you for having me today. I'm looking forward to the discussion.
Excellent. Well, I hope the audience is as impressed as I am about the quality and the combined years of experience on the panel. So for today's panel, we're going to break it down into 3 broad topics, and they are common challenges, future trends, and areas of focus for collaboration. So in terms of common challenges, I think the first one that we'd like to discuss in this panel is ethical, privacy, and data ownership. So we heard a bit about this from the various presenters earlier today. It's evident that the ASEAN region has taken a position amongst the forefront of genomics and precision medicine sciences. And it's part of major international consortia, such as the Global Alliance for Genomics and Health that we heard earlier. However, concerns arise around confidentiality, data ownership, fears of personal genetic data misuse, and questions over de-identification of genetic data. In the UK, we heard there are some remarkable initiatives such as Genomics England, UK Biobank, and more recently, our Future Health Initiative, just to name a few. So I'd like to ask the panel to share their views or learnings that could help navigate these ethical, legal, social policy issues around genomics data and access. And perhaps, Professor Matt Brown, you'd like to get us started on your views.
Yeah, so Genomics England takes an approach of careful prior consent and always bringing our patient participants along with us on the journey and having them at the center of what we do. So our data is only available through a trusted research environment setting. We're looking at having some de-identified summary level data available beyond that. But identifiable data, including clinical data or whole genome sequencing data, is only available within a research environment setting. And we only make available data for which we obviously have full consent. That creates a challenge in that ideally, we'd like to be able to federate data across different research environments and be able to share data globally. And therefore, we're participating in various initiatives, including those run by GA4GH, about how to organize data sharing across research environments. And I think that's a key area for development over the next few years to ensure that these research environments don't end up being silos.
Excellent. Any other comments from the panel? Any other additional points or reflections you'd like to add?
Yeah, maybe it's me, Chairat, from Thailand. So maybe I can share a little bit about this. But as I just mentioned, that we have the Genomics Thailand projects going on running. But at the moment, it's at the starting of the sequencing process. So it's not up to the users or to make use of the genome data we have yet. For sure that for the people that participate in this project, so they need to have the privacy, to have the ethics to keep that confidentiality. And I think for Thailand, we think that what we're going to see from the sequencing project here is to get the information that might be very unique for Thais. And that we have the mechanism that all the data will be anonymous, and we have a sequencing data processing center, data center that can hold everything as the confidentiality as confidential. And this is very important. And I said that to release the personal data is always an issue, that we have to come up with some good ideas and protocol and how we can handle this. I think maybe we can learn more from England, maybe because you have this project so running at the moment. I think, and also I'm not sure about in Southeast Asia or ASEAN countries if they have a genome center and how they do this, and then we can share about this. As I said, that it might be very important.
Excellent. So maybe we can move on to the next topic, and then we can also have some time at the end for any reflections if we want to come back to these. So the next topic is around the integration of the genomic and clinical information and incorporating that into the practice of medicine and our daily patient management. So this integration is inherently challenging, especially for lower income ASEAN countries. The right infrastructure and people power to support it, it's critical in enabling healthcare providers to interrogate a patient sequence and to obtain an informative and accurate clinical report. The scale of the infrastructure required for sequencing alongside large population scale bioinformatics is often available in higher income countries, but often the patients affected by these diseases are based in lower income countries. So how can the UK and the ASEAN region partner to facilitate a more equitable distribution of standardized platforms and skilled talent for more sustained research growth in the ASEAN region? And I thought perhaps Dr. Niranjan Nagarajan, if you'd like to get us started on this particular panel question.
Thanks, Slavi. Yeah, I think it's a very pertinent question for the region. I think, being in Singapore, we face a similar dilemma, especially when you talk to other partners. Obviously, Singapore, with its economic situation, has more resources, but then a lot of the interesting questions scientifically, especially in relation to biodiversity, are in the wider ASEAN region. So how do we collaborate in a fashion where we can do interesting science, but also do science that benefits the people who are vested in that science, or should be vested in that science? I think the fundamental question really is that can we define areas of common interest, and instead of the research being driven by perhaps diseases where there's more money riding behind it, or areas where there's more money riding behind it, we go from more which are the larger populations that are being affected and where things are understudied. So a case in point, in the case of cancer genetics, there are several cancers which are endemic to Southeast Asia, oral cancer and liver, bile duct cancer, where these are areas which are not heavily studied because there are just not enough resources that are being allocated for these studies. By working together and by finding a shared motivation to address these challenges, I think we can divert more resources. So this might be within ASEAN partnerships or ASEAN UK partnerships, where we recognize that this is the insights that we obtain from some of these studies can benefit us all. Another area of really of interest to all of us perhaps is antimicrobial resistance. It affects all of us. So surveillance of antimicrobial resistance determinants and pathogens that are maybe acquiring these plasmids is of interest and relevance to everyone. And the COVID pandemic has really shown the importance of genomic data sharing. So in addition to, in my mind, creating the capabilities, I think us getting all on the same page in terms of our common motivations and our common aspirations, and having forums like this where we discuss those common aspirations, I think that's an important area.
Excellent, thank you. And I see we've got a couple of hands up from our other panelists. Professor Matt Brown, I think you're first.
Yeah, so I just wanted to say the issue of actually translating and enabling genomics within clinical practice is not one isolated to non-first world countries, and certainly it's been a serious issue for us in the UK as well. Part of it, a substantial amount of it, is actually building it into clinical practice pathways and education of clinicians. But there's also a lot to do with just software and particular decision support software. And so Genomics England is working hard with other public organisations to ensure that as much of the software is actually freely publicly available for decision support following next generation sequencing. I think that's a really crucial issue, so that the software is actually affordable to enable the translation in countries which are less well off.
Excellent, thank you. Dr. Shairat, maybe you can finish us up so we can move on to the next theme.
Yeah. I mean, I just want to add briefly about the Genomics Thailand focus area that maybe Niranjan has talked about. The cancer, maybe cancer that's specific to Southeast Asian or whatever. And this is one of our focus area also in cancer, infectious diseases, rare diseases, non-communicable diseases, and pharmacogenomics for operational drug use, whatever. I mean, we have 5 big focus area on Genomics Thailand. So I think from now on, we're going to have a lot of data coming out from the projects, and maybe if we have something that we can talk and we try a teaser, we can connect you with the researchers in Thailand that might be interested to do it.
Excellent. I did see Dr. Mary Miyamoto, you had your hand up. Sorry, if you'd like to add any concluding remarks on this one, then we can move on.
Thank you. I just wanted to make a short comment. So definitely collaboration is very important, and ASEAN countries should learn from the already advanced countries, and also the funding transition coming from outside of ASEAN country, and at the same time, the knowledge and the skill transfer will be the key to the implemented and the catching up to the developed country. Thank you.
Absolutely. I think the knowledge transfer and the shared learnings and so forth are critical to this, and I think there's a great opportunity now. The old saying, we don't need to reinvent the wheel every time. So moving on to future trends. Advanced analytics, including AI, in the genomics market of molecular diagnosis for rare and common diseases. So this one might be quite close to Matt's heart as well. The diagnostic odyssey for many individuals who have disease can often extend to many years, many tests, and with mixed success in diagnoses. However, we know that the speed to diagnosis and thus early intervention often has major prognostic relevance. There are now numerous examples emerging demonstrating early genome sequencing, shortening the molecular diagnosis journey for many patients with rarer forms of human disease. So the question for the panel is, how can academia, governments, and the private sector, who have all got interest in this particular topic, come together to leverage shared knowledge, computation, AI, to generate insights, empower decision-making, and improve and expedite diagnoses and treatments? So I'm happy to open it up to the panel. If you put your hands up if you'd like to speak, and I can pass it around. Professor Brown?
Yeah, hi, Slave. Look, it's a really key issue. Of our 100,000 genomes, 77,000, I think, rare disease families, only a quarter or so have roughly had diagnoses, even though the project started in 2014. And the process of actually identifying disease-causing variants still involves a lot of manual research. So what we think is the way forward is to put the data up in a really good computational facility that's designed to enable AI-based approaches. And we're doing that for both our rare diseases and cancer. This is particularly important for us because we're starting a program sequencing 200,000 newborns over the next 3 years, with a view to newborn sequencing being rolled out as the screening process of choice across the NHS. And for that, of course, we're going to need very rapid diagnosis within a couple of months. And so we're partnering with other global academic institutions and companies like NVIDIA and others to enable this, and it will be both a data and computational resource that will be usable globally.
Thank you, Matt. Okay, Dr. Mary Miyamoto.
I just wanted to also add that our example there, the customer, Stanford University, they recently published a paper in the New England Journal showing the proof of concept, the same day analysis, whole genome sequencing, the partnering with NVIDIA and Google. So that even we have a huge amount of data, but also the AI and the GPU power enable us to analyze it and interpret it and answer the difficult question. That is a really big hope, and we can definitely expect that the more coming from those the rapid turnaround time of the analysis.
And I think is that the Guinness World Book of Records, that one, that now? Yes, I saw that
yes,
one.
we are very excited. Yes, thank you.
Dr. Shaida?
Yeah, I mean, just as briefly about, the question that, how can we, the academia, government, and private sector can come together and leverage knowledge, right? And actually, I can share from Thailand that, in academia, I would say researcher or professor, they tend to be very focused on their research only. And even though they want to translate what they found to the market or commercialization to real use, they need someone to help. And in Thailand, we have private sector and companies that are very good at data processing. They are very interested somehow to work in genomic business. So that's why at TCL we can connect them together, come to talk, to translate the language between researchers and the business partner. I think this is the thing that we have to increase the communication between the private sector and also the academia. And this one we need someone to in between to help them, connect them. And this is what I think we need to have more around the region.
Excellent, thank you. There's a common theme there, I think, in terms of, again, partnership, collaboration, shared learnings across. And I think that's going to come up in all the panel questions and potentially in the next one, which is forward-looking. So, again, looking towards, say, 2026, we all appreciate genomics is a rapidly advancing field. There's a lot of exciting technologies emerging, new techniques, new data science platforms, leading to new biological insights, even sometimes in earlier data generation. And the scale of genomic studies feels like it's really starting to fit an exponential curve in terms of how big the sample sizes are for each new initiative. Among this panel, looking ahead, what prospects excite you the most in terms of these opportunities in the next 3 to 5 year horizon? And maybe I can just extend that with the next panel question, actually. Where do you see opportunities for partnerships to accelerate in this field, and including public health, between the two geographical regions? Are there platforms or events that can accelerate partnerships and collaborations between researchers, business sectors, and investors? Professor Matt Brown, I believe you had your hand up first.
Yeah, hi, Slave. So look, this may come up again, but I would have thought that by far our biggest challenge is to translate what's been learnt in white European ancestry populations into the rest of the world's population. So I think what excites me in terms of having the biggest global health impact is going to be a marked increase in interest in diversity aspects of genomics and genomic medical services. At the same time, it's hard to get away from the fact that genomics technology is accelerating incredibly quickly in development. We've heard on the presentations this morning about Oxford Nanopore and the ability to do long-read sequencing, structural variation detection, epigenetic profiling, all in the same sequence is absolutely fascinating. I think on the cancer front, the ability at single-cell level to look at mutational profiles, transcriptomics, proteomics, and the possibility that that might enable cancer vaccine development, I think is hugely exciting as well, and an area that Genomics England definitely wants to get into. But I'm going to put up front diversity. We haven't talked about polygenic risk scores here, and actually in terms of overall effects on global health, I think polygenic risk scores have a pretty high potential impact as well that we'll see through programs like Our Future Health over the next few years.
Thank you. Absolutely echo the genomics diversity priority. Dr. Miyamoto.
Yes, I'd like to add the genomic diversity, not just the human, and also the variety of the animal and the plant, especially ASEAN countries that are very rich the biological resources, and how to maintain and how to protect from the environment. The genetics information would be very useful. And from this SARS-CoV-2 situation, we learned the power of the genome analysis, and also the open shared information globally, and that can be applied to these bioresource rich the countries in ASEAN, and how to analyze it, and how to share the information, and how to protect the environment for the future will be very, very important. And for that, the accessible the sequencing, and also the not just the centralized location, but the decentralized sequencing, the close to the sample, and the individual get the benefit and see the actual the advantage from the genetic information and genomic resources would be the very exciting time, I believe.
Thank you, Mary. Niranjan?
Thanks, Lavi. So I think I want to extend on what Mari mentioned in terms of going beyond human to plant, but instead moving further into microbial, which is an area of interest for me. I believe there's enormous microbial diversity, more than 1 million species out there that we share the planet with, some of which cause diseases that we have spent a lot of time and energy studying those, but there's immense biological diversity that's evolved over billions of years, and that capability we are just touching the tip of the iceberg in terms of exploiting that capability for various biotechnology applications. So all the way from addressing climate change issues to being able to deliver medicine in a better way. I think there are opportunities here to leverage microbial biodiversity. The CRISPR-Cas9 revolution is a prime example of how we learned a lot from bacteria. So I think this is an area that we can all collaborate and study. There's lesser ethics and privacy concerns when we study microbes.
Excellent. So I think we probably ought to move to, we've got about 7 to 8 minutes left. We ought to move to the Q&A from the audience. We've got a handful of questions that we can go through. So I encourage any of the panelists to just raise your hand if you'd like to address these. And the first one is somewhat related to the recent conversation. So, and it also goes back to Lena's point around interoperability and how that would work between the UK and the ASEAN region to be able to share these data and in a federated manner. So anybody from the panel would like to pick that one up? Matt, you got your hand up?
Yeah, look, clearly federating across global genomics data setups is going to be absolutely critical for, particularly for rare diseases, I think, because so many of them are so uncommon that individual data sets are usually too small. There's certainly in Europe and North America, all of the major initiatives are actually going out of their way to make data publicly available, and I'd hope that actually is something that happens worldwide, and that we don't end up with a situation where we lack diversity because the non-European ancestry efforts don't make their data publicly available. That's something I think we really need to ensure.
I mean, that's a critical point, and if one reflects on what are the barriers to making these data available, there's a lot of layers that we need to get through, and so how can we? Again, it comes back to shared learnings. There's probably a large component of education about genomics research and the benefits to that population, and various other cultural aspects and other things that we need to work through to make sure that it's we get the best breadth of diversity. And I'll move on with that note to the next question, which is very, very relevant. And so I'll just read it out. We all recognize the benefits of diversifying the collective genomic databases to shift away from a very Eurocentric genomics view to reflect the ethnic diversity of the planet. Is there an opportunity with this UK-ASEAN alliance to leverage skills and resources to accelerate this shift? Matt, that's the new hand. Okay, go ahead.
Yeah, sorry, I didn't put my hand down before. Look, there's a huge amount of interest in the UK in, I guess, particularly South Asian, but globally Asian diversity, because so much of the UK population has Asian ancestry. Of course, there's also a lot in African ancestry as well. Programs like Born in Bradford, East London Genes in Health, and Genomics England's own diversity programs are going to be, over the next few years, making great contributions in terms of characterizing common and rare diseases and genetic variation and cancers in these particular groups. And they're all public organizations that are keen to partner with people from those populations around the world. That's not an exclusive list of them. I know that there are others out there that I haven't mentioned as well. But I think, definitely the UK's academic and public organizations are really open for collaboration to make sure that we get over this issue, this weakness in current genomics understanding.
Any other panelists would like to contribute?
Ms. Sharat, can
I go ahead.
Yeah, actually, I mean, so to one of the collaboration that I can tell you about, it's about a program that we call a genetic counseling visiting professional program that TCell has facilitated to work with guys and St. Thomas NHS Foundation Trust, and we connect them to an institute in Thailand. It's Chirapon Royal Academy. So this is to train the genetic counselor. So at least to make use of the genetic, to diagnose or have counseling of the genetic diseases. And this is one of the example of collaboration in terms of clinical treatment that we connect them. And this is one of the things that we still have more other that I think UK and ASEAN countries can connect.
Excellent. Professor John Byrne, I noticed you came on video for a moment. Oh, there we go. You got a hand up.
Yeah, sorry, I wasn't sure whether I was allowed to join the panel, but
we'll make an exception. Go ahead.
I mentioned Global Variome, which began as the Human Variome Project in 2006 in Melbourne, and that has representation across the world, including a significant team in Malaysia. And one of our services or one of our operations is the Leiden Open Variant Database. So we've got about 1 billion variants in gene-specific databases, which can be assembled by curators in any country. And the whole purpose of that, run by Johan Den Dunnen in Leiden, is to allow people to gather the variants of interest to their clinical service. And it's shared anonymously, and it links up to ClinVar, which is now the other major resource of variants of clinical significance. So I think anyone can take part in that. You don't have to be in a big genome center. and that's available. The LOVD database gives access to virtually every gene. There will be information about the variants that are significant in them. BRCA challenge is just an extreme example of that sort of data gathering, which I would encourage people to look at.
Excellent, thank you, and thank you for chiming in. So I think we'll probably conclude there. There are a couple other questions, but I am mindful of time, and I want to make sure Alan has enough time for his closing commentary. I just want to say I hope you've all enjoyed the fishbowl session as much as I and the panel have. I'd like to again take this moment to thank the UK-ASEAN Business Council and UK's Department for International Trade for the opportunity to be involved in the symposium. From my personal view, it's been a true honor to engage as a representative of a company that has dedicated significant attention to advancing our understanding of human disease biology and to find new and innovative ways to treat, prevent, modify, and one day hopefully cure diseases for patients across the globe. With that, again, thank the panelists for your time and your expertise, and wish you all the very best as we approach the close of this meeting, and I'll hand it over to Alan.
Slave, and to our wonderful panel, thank you very much for that highly impressive discussion on how the UK and ASEAN can collaborate. We have heard today the depth of work and research underway in both the UK and Southeast Asia, and we hope that you have found today's symposium inspiring. Given the many benefits that advancing genomics brings to our communities, we look forward to seeing a greater partnership and alliance between the UK and ASEAN. That brings a close to today's symposium. Today's presentations will be sent via email along with a YouTube link if you'd like to watch this symposium again or to encourage your colleagues to watch. On behalf of the UK government and the UK-ASEAN Business Council, as well as the ASEAN Business Advisory Council, the Ministry of Health Malaysia, and MyDigital, a very big thank you for joining us. Enjoy the rest of your day. Thank you.
Bye.
Thank you.